I would just like to start by giving you an introduction of where I work and what we do. So let me just have the pointer. There we go. So I work at the Centre for Brain Research at the University of Auckland at the Neurosurgical Research Unit. So this is a research unit that was established by my bosses here, Professor Mike Dragonoff, Sir Richard Fall, Professor Richard Fall, and Professor Morris Curtis. And really it's to have this entire circle where we work with the patients and the clinicians at Auckland Hospital to receive tissue from neurosurgical specimens and to do that research at the university to find new treatments so that hopefully we can circle that back to potentially help the patients that are suffering from those diseases. So these are the main neurosurgeons that we work with, but we work with the whole sort of entire neurosurgical unit at Auckland. So Mr. Mee, who was the head of department at the neurosurgical unit, Mr. Schweder and Mr. Correa, who are the neurosurgeons there, and we work really closely with them on all these projects. And of course, I can't leave out the talented and hardworking postgraduate students. And a lot of the work that I'm going to present today is going to be their work. And you can see this was before COVID, and this is during COVID. So most of our meetings were virtual, just like today. Now, on the topic of brain tumors, the Neurosurgical Research Unit over the last 7 or odd years, we're really grateful to be able to receive about 200 brain tumor specimens. So these have all been housed, and over 60 of them are actually high-grade glioblastoma cases. So all these tissues are fixed for histological examination. Most of them are frozen for molecular studies. Many of them have been cultured, so they're kept alive as cells for studies, as you will see. And a lot of the patient data and MRI, which are all anonymized, are also collected so that we can do some analysis on the results that we get compared to how the patient's MRI and data, the clinical sort of history went along. So I don't think I need to introduce too much about brain tumors, so I'll be very quick. So obviously brain tumors are largely separated into primary and secondary. Being a primary tumor means it actually the tumor arose from brain matter, so something in the brain. While secondary tumors tend to be where you've got tumors from the periphery, so other parts of the body that ends up metastasizing into the brain or the spinal cord. Now, the grading system is constantly changing, and this is always going to be updated. And the most recent iteration came out in 2021. And the real take-home message is we're now adding more and more molecular features. So it used to be more histological and then a little bit of radiographical. And now it's a lot more in terms of molecular profiling that is getting added to make it a little bit more personalized, shall we say, in terms of tumor diagnosis. Now, we're going to largely focus on the highest grade brain tumors, which are GBM, but a lot of the research that I'll be telling you about could also be applied to perhaps other brain tumors as well. The glioblastoma, as you know, is probably the most common and aggressive malignant brain tumor in humans. Now, its median age is over 60. It has a higher incidence in males and or those of Caucasian descent. We really want to know the New Zealand incidence, yet there is no central brain tumor database yet. And this is something that we're trying to work together with Chris and his team at Brain Tumour Support to really see if we can get this going in New Zealand as well. And as I told you before, it was historically largely diagnosed using histological examinations, where they look for certain cell types, how fast the cells proliferate, and certain features such as necrosis. But as mentioned, it's increasingly assisted by genetic diagnosis. And this is not to confuse you in any way, but this is a nice diagram showing how these molecular features are now being used to separate the different tumors into its main groups. So it used to be where all these grade 4s down here were largely grouped as a single tumor. Now they're getting quite separated into each individual sort of tumor types. And that will definitely help with future prognosis, but also to develop treatments for each sort of specific type of tumor. Now, as you know, with the current clinical practice for glioblastoma is if they can, they will do a debulking of surgical removal unless the tumor is in very delicate areas or dangerous areas for surgery. Then they undergo adjuvant chemotherapy, temozolomide being the main chemotherapy agent, and radiation. And this has been dubbed the Stubbs protocol. And that's because this gentleman here wrote this landmark paper that showed this for the first time, or properly for the first time in a clinical trial. And then, but even with this, most of the GBM tumors almost always reoccur, unfortunately. And the overall median survival time post-diagnosis is still around 15 months. This is still 3 months more than before Stubbs protocol, but we haven't been able to improve on those 3 months over the last 17 years. And we're trying really hard, the community is trying really hard to see if we can rectify that. Now, an important factor here is this MGMT methylation status. And I'll show you briefly what that means. So if you see this graph here, this is a probability of overall survival graph. And you can see that tumors that have an unmethylated MGMT, so just without going into any detail, those two, red and blue, means that the MGMT gene is active, it's on. And when that gene is on, so if you see here, that nullifies the effect of temozolomide. So temozolomide normally puts a methyl group onto this part of the DNA, and that if it stays there, that causes the cell to undergo a futile mismatch repair cycle, which in essence should lead to cell death or cell senescence. And that's sort of how temozolomide is shown to work. If this gene is active, which is the case in this red and blue patients, then they repair this. So that basically renders temozolomide almost inactive in a sense. And you can see that the patients do considerably worse. And if you have it methylated, so if this gene is turned off, then you're not going to get that repair. So temozolomide has a greater effect. So this is a major problem with temozolomide, I guess, is that it is effective, but if you have a certain genotype, shall we say, or molecular phenotype, then it's not so. And this is where the current molecular analysis of GBM is really coming into fruition, really, is being able to tease these things apart. Now, if those don't work, so on resistant or recurrent GBM, then there are some second-line treatments. So BCNU or CCNU, things that are often given in New Zealand, at least, and mostly around the world as well. But they haven't been as effective in extending overall survival, shall we say, and it's given to patients that do show resistance. So there isn't a lot of other choices out there at this stage. Now, what has advanced more recently, as many of you know, bezimizumab, which is Avastin, which is an anti-angiogenic. Well, when I say drug, it's more of an antibody. And as many of you know, it prevents new blood vessel formation. And it's worked in other tumors. And therefore, they thought, okay, brain tumors, especially GBM, has a lot of new blood vessel formation that feeds the tumor. It should work really well. And they tried this. And they found that there was a prolonged progression-free survival, meaning it took longer for the tumor to come back. But they did see, or they failed to see a huge benefit in terms of overall survival on its own. But what other things have advanced since then is this non-invasive device called tumor treating fields, where you have a electrical field, alternating electrical field device on the patient's head. And this creates an electric field that disrupts cell cycle of fast dividing cells. And in the brain, the fastest dividing cell should really be the brain tumor cells. So it tends to affect the brain tumor cells more than other cells. And when given together with temozolomide and radiation, this has been shown now in clinical trials to extend overall survival time by over 5 months if it was adhered to, and the 5-year survival rate went up to 29% in patients that really adhered to the whole protocol. And what's interesting and what's exciting now is that when they started combining these other therapies along with the tumor treating field, they're seeing even more benefits together. So those drugs that were not as effective on their own are showing better progress and outcomes when they're being combined, which is really encouraging. Another hot topic, which is under a lot of research, is immunotherapies. Now, first, our vaccines. Just like our COVID vaccines, what these vaccines do is it allows our body's immune cell, the patient's immune cell, to be able to recognize the tumor antigens. So things that are expressed pretty much only by the tumor or a lot more by the tumor. You vaccinate the immune cells against those proteins, and then your immune cells will go and attack the tumor. And the idea is that it'll end up killing your own immune cells end up killing the tumor. Now, there are peptide-based, so these are just protein-based vaccines, but also something that's quite interesting, and a new clinical trial recently came out with results, is dendritic cell vaccines, where they actually removed the immune cells from the patient, and then they harvested, and then outside the body, they then exposed them to the actual patient's biopsy tumors, so the tumor tissue. And therefore, these immune cells learned outside what the tumors are made out of, and what things are expressed by the tumors. And they form some kind of a response to those tumors, and they were injected back into the patients. And they were basically given a license to go and kill the tumor. And they've now shown a modest increase in overall survival, as well as 5-year survivals recently. But there's a little caveat to this. Their controls were not an internal control. They had, for certain reasons, they had to use controls from other studies to compare. So not a perfectly controlled study, but still very encouraging indeed. And other immunotherapies include adoptive T-cell therapy. So T-cells, as many of you know, are the immune cells that will go actually and attack the tumor and kill it. The problem with GBM and a lot of the high-grade tumors are the T-cells are not really getting into the tumor. So it's GBM, or these high-grade tumors, are known as cold tumors. And that means these immune cells are not really getting in there, or even if they do, they're getting turned off or turned down. And one method that's worked really well in peripheral tumors is this chimeric antigen receptor T, or CAR T cell therapy, where they're using genetic engineering to genetically engineer certain T cells to recognize tumor antigens, so brain tumor antigens. And they're showing some promising results, but one of the problems with brain tumors is there aren't many well-known brain tumor antigens. So things that only belong to the brain tumor. There's not a lot of molecules that are known that we can actually target, and that's sort of been part of the problem. And they're trying really hard to find more tumor-only antigens, and then being able to literally teach these cells or make these cells recognize the brain tumor. Because once they can see it, they can go kill it. Now, another one is checkpoint inhibitors. And these are immune checkpoint molecules, are molecules that are naturally in our body. They're always in our body. And their role is to tone down the immune response after it's been elicited. Because we don't want our immune response to continue. You want our immune response to occur, do its effect, have its effect, and then get turned down again. So it's like a little balancing tool. The thing is, brain tumors use this against our body. So they express a lot of these checkpoint molecules. And when an immune cell comes in to kill it, it actually turns it down. It actually turns it off. And that's been a major problem with immunotherapies for brain tumors. And that's why it hasn't worked so well compared to many other tumors so far. And another thing is that there are molecules that are known to be able to turn off these checkpoint molecules. So when checkpoint inhibitors are used in combination with the above, they've been seeing that they think they're going to see a much greater effect, and those trials are being done as we speak to see if we can combine these together to get a greater effect. Now, however, and this is where our study sort of starts, as you can see from what I presented, most of these treatments for brain tumors have really adapted therapies that were developed for other tumors. So they're using therapies that were developed for melanomas or lung or breast cancer, and they're applying that to our brain. And that's absolutely fine because brain tumors are still tumors and they share many similar features to other tumors. However, our brain has a very unique environment, microenvironment. Our brain is very different, as you will see throughout my presentation. And as a neuroscientist, so my background is not a cancer biologist. That wasn't my initial training. My initial training was a neuroscientist. So I really wanted to tackle GBM from a neuroscience perspective. So from the brain in rather than from the tumor out. So some of the projects that we are undergoing in our lab are these, and I'll go through each one. So I won't read through this slide right now, but these are what's going on in Auckland as we speak. So project one is looking at GBM cell migration. So one of the features of glioblastoma, these high-grade tumors, is the fact that they actually divide really quickly and they move throughout the brain, and that's what makes it extremely difficult to treat. As you can see, some of them cross the midline, and that's why sometimes they're known as a butterfly tumor, because it almost looks like they're spreading out their wings like this onto the other side. Now, one of the molecules that I had studied when I was doing more neuroscience and brain development was this molecule called PSA-NCAM. Now, this molecule, long story short, if this molecule gets expressed, the brain cells can move a lot easier. Because if they don't have that molecule, so this red molecule is PSA-NCAM, if they don't have it, the cells are stuck in place. So they will stick and they won't be able to move that easily. The moment you make this PSA molecule, the cells become slippery and they're able to move through the brain. And this is how our brain develops. When your brain is developing as a young person, in utero as well as a young person, new cells are being made and your brain's growing. And this is what allows cells to move. And even as an adult, as we learn new things, there's new neurons being produced. That's a big theory. And in doing so, this molecule gets upregulated. Thing is, because these are involved in migration, people have also found that it might be involved in tumor metastasis. So any cells that are moving might be utilizing this mechanism to their advantage. So this is where we started our project off, where we looked at this molecule, and I'll just show you this study here. This is the PSA-NCAM, and you can see that if you get rid of that PSA-NCAM here in red, so you can see red there, there's nothing here, you basically stop the cells from moving. So you can see that you can stop the cells from moving, and they basically get stuck in their place and differentiate. And if we can do this to brain tumors, then we can stop them from moving from where they have started, and that'll make the treatments a lot easier. So we went through and looked at 98 brain tumor specimens with collaboration with Professor Maurice Curtis and Mr. Peter Hepner, who's the pediatric neurosurgeon in Auckland, and Dr. Bernard Kim, who's a clinician at Auckland Hospital. And we looked at the tissue. And here we use Ki-67, which is a proliferation marker. So it looks at cells dividing. And this is what the pathologist will use to grade tumors. So it's a gold standard in terms of pathology and tumor grading. And then here is the molecule PSA-NCAM, which is the migratory molecule. And when we looked at the tumor sections, the brain tumor sections, you can see grade 1, grade 2, grade 3, and grade 4 being GBM. As the grade increases, you can see that the cells have more proliferation, which makes sense. This is how the pathologist will also grade their tumors. It's one of their criteria. And you can see as below, PSA-NCAM load gets higher and higher as you go into your high-grade tumors. So showing perhaps that PSA-NCAM might be involved in the migration of the tumor. When we then looked at, married this up with patient outcome, we found that the tumors that had, the tumor tissue that had less PSA, the patients did a lot better in terms of overall survival. Tumors that had high PSA load, the patients deteriorated a lot quicker. Now, this was almost, this wasn't unexpected because, as I mentioned, the lower grade tumors tend to have less PSA, so lower grade brain tumors, the patients will survive a lot longer. So this was not unexpected, but it was nice to see. What was more interesting was when we only looked at the high grade brain tumor, so just GBM. Within GBM, there's still patients that show a low PSA versus high. And when we just looked at the cell proliferation in the high grade tumors, we could not separate the patients out as a cohort. But when we looked at PSA, even in the high-grade GBM, patients that had less PSA, which is this blue line, survived a lot longer than patients that had high PSA in their tissue. Again, not directly telling us whether this is the cause, but at least correlating the high PSA load with patient outcome. And we did this through univariate analysis, so the survival analysis, multivariate, and they all show that PSA-NCAM had a very high hazard ratio, meaning if you had high levels of PSA, you were 2.4 times more likely to have a worse prognosis. And this is just looking at genes for certain types that actually put on PSA. So all in all, showing that having high PSA was not a good thing, and we're trying to find out what it is doing to our tumors. And just to see if we can get any MRI signatures, so radiographic signatures, so that it's non-invasive, we also collaborated with engineers as well as radiologists, and we found that tumors that had high PSA also showed higher volume tumors. So the tumors were getting bigger, or tumors were bigger if the patients had high PSA load. So this sort of set the scene where, okay, patients that do worse have high PSA in their tumors, they tend to, what are they really doing? Now, this is what the heading here, treatment-resistant GBM cells. This is another reason, another root cause in literature and in academia that we think GBMs are so hard to treat. We have these, GBMs have these glioblastoma stem cells or initiating cells. And these cells are stem cells and they hide in these hypoxic niches in our tumors. And they tend to be a lot more chemotherapy, so temozolomide and radiotherapy resistant. And we've shown that in our lab as well, where if we actually give these patient tumor cells temozolomide and even radiation, we shoot radiation straight at them, it doesn't kill them. So they actually survive this. And as you know, these are the current therapies that we are actually giving. Therefore, these stem cells might be actually surviving. And how is this related to PSA? You'll see, I'll show you now. Through our collaboration, we will receive donations, kind donations from the patients of the brain tumors, and we can actually grow these at the Hugh Green Biobank at the Centre for Brain Research and keep these cells alive for experimentation. And these cancer stem cells can be grown. These GBM cancer stem cells can be grown. So here is the initial isolation. You can see it's a bit of a mesh because all the cells are being isolated. And then over time, within a week or so, they grow into these balls called tumor spheres. So they're made out of the stem cells. So the stem cells are growing as these balls of cells, as if they're almost as if they're growing like a tumor inside the brain. They grow as a ball. And when they do so, we can plate them out and they actually move and we can actually see them migrating. So I'll show you this video, hopefully it plays. So you can see that from that ball of tumor cells, those GBM cells, these stem cells, we can actually literally film live how these guys are moving away, sort of mimicking how the brain tumor might be spreading in the brain. And lo and behold, these cells that are spreading express high levels of this PSA-NCAM, the same molecule that we saw in patients' tumors, in patients that did worse. So In migrating, this molecule might be quite important and could be a vital molecule in instigating the migration of these brain tumor cells. So what we have done is we've used certain enzymes and drugs to see if we can lower this PSA-NCAM in our patient samples. And you can see here clearly the green is the PSA-NCAM, and you can see drug A and endo-N, which is an enzyme that cleaves this, gets rid of this PSA-NCAM quite a bit, reduces it. And when we do so, what we found through live cell imaging is that the migration has been stunted quite a bit. And we can actually follow each cell and their migration, and you can see that they're migrating much less if this molecule has been removed. And we can see that there as a migration analysis. So with all this, we're now trying to put together what is the mechanism of this migration? Why is it decreasing? And what might also be increasing this migration in terms of therapies that are going on for brain tumors? One of which is radiation therapy. Now, if you remember, the tumors that are getting radiation, almost every brain tumor gets radiation therapy. But those cancer stem cells were the ones that survived. And what they found, and many studies found, that when the tumor cells survive the radiation, they become a bit more migratory. So they turn into a more migratory cell, which is not what we want. So we started this project along with radiation specialist and cancer biologist, Professor Wilson, Dr. Hong, and Dr. Han, who is a consultant oncologist, and 2 graduate students, Ramika and Imogen, that are working on this project. And we decided to look for whether radiation really do increase migration. So this is some data from our lab showing that yes, if you look at this graph here, if you give tumor cells radiation, the cell numbers do decrease, but they don't all die. So there are these surviving cells. And those surviving cells actually increase PSA-NCAM, as you can see here in green. So they actually overexpress it. And it's hard to see here, but they actually migrate further during the time frame that we've filmed them. So radiation might be increasing PSA, and that might be causing them to migrate more. Therefore, if we can find ways of decreasing this PSA or other migratory markers, then we might be able to restrict the cell migration that is induced by radiation. By keeping the cells in one spot or in the proximity of where it's being radiated, we'll have a lot higher chance of being able to kill these cells through multiple doses of radiation. So this is the idea behind this study that we are currently conducting at the CBR. So in summary of the first project, I showed you that in patient tissue, we found that high PSA was related to high volume of the bigger tumors and worsening prognosis. We could culture these cells in our lab from patients, and we could see PSA-NCAM being expressed in the migrating cells. And then we found that radiation might be enhancing this, and we could potentially screen for drugs that could decrease this migration. So that's the summary of project 1. A second project that we have in our lab is more to do with the tumor microenvironment. So the tumor when the cells come into the tumor, we've got many cells that the tumor cells interact with. And that's what we call the tumor microenvironment. And we've got a group of talented postgraduate PhD students that work on this project, as you can see there. And the aspect that we want to first look at is the immunosuppression. If you remember back to at the start, I said GBM tumors tend to suppress the immune system. Therefore, it makes it harder for our immune system to kill these tumor cells. And just quickly going over to the blood vessel, this is the first entry point of most of the immune cells. Most of the immune cells will come through the blood into the tumor. So the blood vessel will be the entry point. And just showing you this, our brain has a lot of blood vessels. It's one of the most well vascularized organ in our body. And if we actually sum up the length of all the blood vessels in your brain, we can basically go from Auckland to Wellington and a bit more. So that's a lot of blood vessels and a lot of entry points into our brain. However, many of you know that the blood-brain the blood vessels in our brain have this thing called a blood-brain barrier. And I say it should really be called the blood-brain customs gate because it's not like a full barrier. Yes, it blocks about 98% of molecules that could actually come in, but it does selectively allow molecules to enter the brain, just like the customs gate at Auckland Airport. So this is sort of a schematic or illustration of the blood-brain barrier, and there are many cells that make up that blood-brain barrier. One of which is this green cell that I've highlighted called pericytes. And what studies have found is these pericytes are these gating cells. So when things are trying to come into the brain, these pericytes are there to greet them, and they could also convey messages from the outside into the brain and might be involved in the immune system or the immune microenvironment of our brain and potentially our tumors. So this is work conducted by Max, who's now a clinician, and Jenna and Susan in the lab. And their work looked at these pericytes, these cells from patient tumors. Now, sometimes when the tumors are deep inside the brain, the surgeons have no choice but to cut out a little bit of the normal brain to access the tumor. It is inevitable to cut out a little bit just to access to debulk the tumor. And in those cases, we're able to gain this very precious resource where we have some normal brain and tumor from the same patient. And therefore, we can actually compare directly the two cell types. So here you can see these are the gateway cells from the normal brain, and this is from the tumor from the same patient, and we can compare them side by side. And without going into too much detail again, what Jenna found was that the cells, when the cells from the normal brain from the same patient were stimulated with an inflammatory molecule, so something that will cause inflammation and immune system response, they responded really well. So these cells are known to respond really well. But in GBM, can you see that they have a much more dull response? So this is sort of showing us that perhaps these cells are actually playing a part in dulling, like lowering the immune response. So here, just summarizing, the GBM perivascular cells could be contributing to the immunosuppressive microenvironment. They may also be secreting things that will be helping the tumors grow. And we could actually find druggable targets on these cells that could potentially reverse this immunosuppressive nature of our cells. The next cell type that we'd like to look at is the actual immune cells themselves. Now, our brain tumors are made up of largely two immune cell types. One, the immune cells that are actually already in the brain. We have our brain's own resident immune cells, sort of like the security guard of a building, they're always there. And then when the tumor starts to arise, you get recruits coming in from the blood. So we've had this two population all mixing in the brain tumor. Now, they used to actually group these two cells as one and try to target these cells as one. But we found that this should not be the case. And we started a study to see if these two cells, microglia, which is the brain's own immune cells, and tumor-associated macrophages, these are the cells that came in from the blood, were actually different and were having different functions. Therefore, we should be targeting them separately or individually. So there were studies showing that perhaps they had different functions. So we started the study to interrogate these two cells separately. But until we started the study, it hadn't been done in human tissue. So we were the first to really separate these out in tissue. And this was done by our graduate student, Zoe, and also in collaboration with Dr. Molly Swanson. And we found molecules, and just to guide you in this picture, this is a brain tumor, and you can see the cells in green are the cells from the outside. So these are immune cells that came in from the blood. You can see we have a lot of immune cells that are in the tumor. They're just not killing the tumor cells. They're just there, idling. The ones that are in red or orange are the ones that came in from the brain. So they're the own brain's immune cells. So some came in from the brain, some came in from the blood. And you can see here, if I guide you down to this bottom, the key takeaway of our study was our brain tumors show clear two populations. You can see there's a population up here and a population down here of our immune cells. Well, if you just look at the normal brain, you only have one population. If you look at meningiomas, which is another type of brain tumor, or low grade, you really only have one type of immune cell, but GBM and higher grade tumors have two, meaning we might need to target them separately. And that's what we've also shown with patient outcome, is that if there was higher brain immune cells in our tumors, so the microglia, if you had more of the immune cells that came in from the brain, the patients tended to do better. So perhaps the security guard of our brain is actually trying to fight the tumor while the blood recruits are not doing their job very well. Now, this is still up for debate. This is all tissue-based studies. We're now at the stage where we're trying to look for functional studies. Are they really doing this? And this is work that's ongoing, so I'll probably skip this slide because it's a little complicated, but Zoe is now moving on. She has handed in her PhD and she's now moving on, looking at how different regions of the tumor might be affecting these cell functions, along with all the collaborators there. So the summary here is that GBM tumors are comprised of two distinct immune cell types, one from the brain, one from the blood, and these appear to have different functions and be influenced by different environments in the tumor. And therefore, we need to target these perhaps separately when we're designing drugs. And this leads us to the exciting part of our talk, which is really developing GBM drugs. And this is work that we started with the medicinal chemist at the Cancer Society, and really led by wonderful students. So Liz, who's been a fantastic PhD student that has led much of this work, Anna, who joined us for a year for honors and did a lot of the work, and Karina is now just joining us as a PhD student, continuing on this work. And the idea here is that we're going to use, since we have the stem cells, the cells that actually survive all the treatments from all the patients, we're going to test a lot of our new drugs on patient tumor cells. And only those drugs that get screened on the patient tumor cells and show effectiveness are going to be investigated further and then taken into animal studies. This is a huge advantage over many other studies where they don't actually look at patient samples, but they look at commercial cell lines. And the commercial cell lines do not have the same variability as patients do, because as we know, we are all different, and it's actually important to screen on actual patient cell lines, and that is our belief. So the signals that we've been largely targeting are mutations that happen in GBM. But the thing is, these mutations are not only unique to GBM. Many of these mutations and signaling, aberrant signaling, also happen in normal tumors. So therefore, there are drugs already FDA approved for many of these tumors. The problem is when they did clinical trials for GBM or brain tumors, they had no effect or very little effect. So the trials failed. One of the big reasons was the drugs could not get into the brain through that blood-brain barrier, or they were spat back out by the tumors through efflux pumps. So that's been a big problem, and that's what's quite unique about our brain. And therefore, we collaborated with these medicinal chemists, Dr. Jenny Josie and Peter Choi at Medicinal Chemistry, and they had this dye that was known to actually cross the blood-brain barrier and enter the tumor. And we published a paper showing potential mechanisms of how these drugs might be entering the tumor and getting into entering the brain and getting into the tumor cells. And just very briefly, as you can see here, these molecules get transported up through these transporters, which are in green. And you can see here in patient tumors, it is absolutely loaded with this green transporter. Therefore, it's taking up this dye, while a normal brain has very little of these transporters. So it's going to largely target tumors. And what we found is that our patient cells also have that. And Liz did a really cool experiment where she actually increased the transporter, and you can see there's more drug. Red is the drug. You have more drug when you increase the transporter, and if you block the transporter, there's less drug going into the cell. And therefore, the chemists did a lot of magic, and they added actual anti-cancer agents onto this dye to transport. And we screened a bunch of them, and we found a lead compound that was really, really potent in killing the patient tumor cells that we have in our labs. And here you can see that's a huge left shift, meaning they become a lot more potent in the nanomolar particle range. And we compared this against all the currently used and trialed drugs, and we found that it was still much better than the drugs that are currently out there. And that leads to why I'm here in Korea, because we now did all these studies and we found the lead compound. We had to then test on animals, and we found this collaborator that had all the right devices and skills to look at our drugs. And here, this is us in Seoul, and we are now actually creating a brain tumor model of a mice. And just as you can see, when the drugs go in, the drugs actually only localize to the area where there's the brain tumor. So it's actually getting in and now getting into that brain. And right now, what I'm doing as we speak is we're trying to see if the animals actually survive longer and the tumors actually shrink. So that's me in the animal room right now, a couple of hours ago, doing all these studies. So, and I'll skip this last part, but the part that I really want to show you is sort of the vision that we have in our group. So as I showed you, we actually we collaborate with the hospital and receive the patient tissue, and we can do cell work and tissue work to look at the biology. Based on that biology, we work with chemists to design drugs. We test those drugs, and if they look promising, we do animal studies to take that a step further. And what I sort of skipped towards the end, because I know I'm going way over time, is we are now computing that onto a machine learning database to be able to use all that information to facilitate precision medicine. So a patient-based medical system that is based on artificial intelligence and actual experimental data. So that's a project that's currently going on in our group. And this is sort of our group's vision to hopefully make this a platform that allows for a lot more precision medicine. So just to acknowledge, I would really like to acknowledge all the patients and families for donating their tissue for research. It really can't, none of this can really work without them. And also I started to appreciate while I was doing animal work, all the sacrifice of the animals for the gain of humanity, I guess, for mankind. My mentors, the 3 professors and a sir for all the mentoring and support, all the graduate students who did pretty much all the work that I presented, and all my collaborators that I work with. It's a huge team effort: neurosurgeons, neuropathologists, oncologists, radiologists, cancer biologists, chemists, engineers, neuroscientists, and here in Korea as well. And all the supporting funding bodies that funded my work and still funding my work as we speak. So sorry about the long talk, I've gone way over time. Sorry, Chris. Thank you very much. And this is my contact if you have any questions, and I'm happy to take some questions. Thank you.
Well, Thomas, no need to apologize. That was a fascinating talk. And before we get into questions, can I just say a comment that I did not know that we had so many smart, clever, bright young people researching brain tumours in New Zealand. And that's really an encouraging thing for me and really a source of hope, because, you know, the studies that you've described this evening hopefully will translate in the future into better treatments for patients. And it's just fantastic that to know that that is going on in New Zealand. So we do have time for questions. So you didn't go on too long. I don't see any questions in the chat. I've got a couple of questions myself, but maybe there were some questions sent in before your talk started. So if I just go over those, perhaps. First one was from Kirsty. Are you looking into genetic links with brain tumours, particularly glioblastoma?
Right. Yes, I mean, as we mentioned, the glioblastoma, there's an increasing emphasis and push to look at genetic subtyping of glioblastoma. And I haven't mentioned everything yet. I didn't mention everything in our presentations, but there are many subtypes from academically that we group tumors on based on their genetics. Yet we haven't really found one thing that has worked for a genetic phenotype yet, perhaps because GBM is a heterogeneous disease. What people have found is even within one GBM from a single patient, there might be several different genetic types of tumors going on. And therefore there needs to be work needs to be done, but there is a lot more work going on this, as I mentioned. And there's a whole field called pharmacogenetics, which is basically looking at how genetics affect drugs going on as well. So that's a field that is really growing as we find out more about GBM and brain tumors and their genetics, it's only going to get higher, like more. It's only going to expand. But at this stage, we don't have a specific marker or genotype that is attacked by a certain drug per se, other than your IDH1 mutation and your MGMT, which is already known.
Yeah, and just to follow on from that, what about in terms of hereditary genotypes? Because a lot of people want to know that if they have a glioblastoma, are their children at a higher risk of getting a glioblastoma, or?
No, I mean, there hasn't been a clear genetic link for sporadic glioblastoma that happens in older age. So your typical 50s, 60s glioblastoma. The childhood glioma or aggressive gliomas, some of them have a genetic component, but that's not sort of grouped as a glioblastoma as such. But it's more NF1 or neuroblastomas might have more of a genetic component. However, the adult GBMs, there hasn't been a clear genetic link that could be inherited as such, that has been well documented. Of course, there are studies that show correlation, but there hasn't been a single one that has been clearly delineated for such a thing.
Yeah, it's interesting because we do hear of cases of familial glioblastoma, you know, having a tumor in the family, but as you say, there's no proven link yet. I've got a question here from Peter, who wants to know about, and you mentioned methylation status. I guess they're talking about MGMT methylation and effectiveness of Avastin or bevacizumab and irinotecan. Do unmethylated tumors respond better to this therapy than they do with the temozolomide and radiation?
Yes, I guess for this, they've done a few combination studies from what I know, and they've done a phase 2, if I am correct. And on that, they found that the progression-free survival might have slightly become a bit better with that combination therapy, but they couldn't really see an overall survival benefit. Again, what they have done though since then is they've done a much smaller retrospective study, and I sort of mentioned that during my talk, where they did this combination basically with your tumor treating field, the device that you had on your head. And they found that had a much better prognosis with that. And they're doing a larger trial because that was a more retrospective study. So people that had that combination therapy done, then the TTF, tumor treating field, became available. So they did that on top of that, obviously, because the tumors were not receding. And so they did a retrospective study of the patients that did that. And they found benefits. So they're seeing if they can do a proper clinical trial on that combination now. So using tumor treating fields along with that combination.
Yeah, that's interesting. Unfortunately, we don't have tumor treating fields available here. And and I hear it's very expensive. So unless our government are going to pay for it, I don't think it's going to come. That brings up a couple of questions asked by Les. He submitted these before your talk. He makes the first one as a comment. Can you please join us and lobby Pharmac to wake up and fund Avastin? So I think there'll be a lot of people here nodding their heads and shouting yes to that, because that is I know it's off topic for your talk, but there are people who are self-funding drugs like bevacizumab, and Les, I guess, wanted to make that point.
Yes.
His other question is, what if any new treatments are there for grade 4 glioblastoma patients? And I think you covered those in your talk very well. Maybe if I could phrase it this way, do you think of the treatments that are currently being researched, do you favour any one or two of those that might be approved in the future? What are the most promising ones, do you think?
Right. Well, going back to sort of the comment on Pharmac lobbying, I mean, as any cancer researcher, especially brain tumor researcher, will tell you, because of the lack of current treatments that are available and the severity of the disease, we really do need to try everything possible. So yes, I'll be absolutely, if I can, I don't know how much help I will be, but I will pull any weight that I can to support expanding the therapeutics for the treatment of high-grade brain tumors, for sure. So just let me know how I can help, and I will do so. In terms of treatments that are going on, I just from what I can see, I think the combination of the immune checkpoint blockers or inhibitors, along with some immunotherapy, because it's worked so well in peripheral tumors, might be. Now, getting those antibodies and those molecules into the brain is another whole nother topic, and people are trying, and they're really cross-fertilizing a lot. So they're using ultrasound. I didn't mention that, but there's often using ultrasound to open up the barriers to allow these molecules to also go in. So there's a lot of cross-fertilization happening, and I really think soon there might be one of those trials where they actually put all of these together for treatment for GBM, because a lot of the actual technology is already developed. It's a matter of putting them together, just like the TTF, where they had these separate trials, and then somehow they serendipitously basically did that together, did a retrospective study to show that it had a big benefit. I think these coming together will have a huge effect. Now, of course, drug delivery into the brain, like the ones that we are doing here, might also have huge benefits soon, because we're already using drugs that have FDA-approved functions for tumors, and it might be still targeting the same pathways that are in GBM. So those also look promising. It's just a matter of how much can we get those drugs into the brain without getting it spat back out, and then testing that through. So those are, in my view, a key area for brain tumors. Other than that, yes, as you would see, if you look at the trials that are going on at phase 2s and 3s, there's a lot going on right now. So fingers crossed. It's impossible to know every trial, but there's a lot going on, and there's more going on. And every time I go to a brain tumor conference, there's more people studying brain tumors from different fields. It used to only be oncologists and neurosurgeons. Now neuroscientists are coming in, like us. And there are other fields, engineers, and more, the diversity is growing, and I think it's only going to get better. We really need cross-fertilization.
Oh, that's great. Yeah. And I can see with the research projects that you're supervising, the one with parasites and the drug conjugates to cross the blood-brain barrier, I can see that's an area that you're really focusing on, so that's great. We are running out of time. It's just ticked over 8 o'clock. There's a couple of questions in the chat box. If we take those two and then finish up, would that be all right, Thomas?
Which one would that be?
Actually, I think there's one from Pete. Pete, if you're still there, did I ask your question already about the?
I think that's been asked.
Yes. And then that's great. If I don't phrase your question properly, please just speak up and intervene. And the final one from Alan, and I think you've just mentioned this Keytruda, which is pembrolizumab, which is a checkpoint inhibitor. Alan asked, does Keytruda have a role with brain tumours? And I think you've answered that one already too, Thomas. Have you got any other thoughts on checkpoint inhibitors?
No, not at this stage. But as I mentioned to you, Chris, at the start, if there's other questions, please feel free to email those to me, and I'll try my best to the best of my knowledge. And if it's not in the field of my specialty, you saw our team of people that are interested in brain tumors, all the clinicians and other engineers and chemists. I can always pull on them to answer those questions for you, so that I can give you the most informed answers back. So I think that might be the best medium. If you have any other burning questions, I'll be able to get back to you that way.
Oh, that's great. Thank you for that, Thomas. And can I just, on behalf of everyone here, thank you again for your presentation. You've obviously taken a lot of time to prepare it and also to deliver it from South Korea. So I hope the timing wasn't too bad for you.
No, not at all. It's 5 o'clock now here. It's 3 hours behind.
Right. Well, we're a couple of minutes past 8 o'clock here, and we scheduled to go for an hour. So I think we'll end this meeting now. And just again, on behalf of everyone and behalf of Brain Tumour Support New Zealand, thank you very much for your talk.
No, thank you very much.